Hereby I open this academic ceremony at Maastricht University. My name is Ton de Goey. I am the Prorector of the ceremony and Chair of the Degree Committee. Welcome all members of the Degree Committee, and then I would like to welcome in particular the three supervisors. First of all, Professor Uli Schotten. He is Professor of Physiology at Maastricht University. Professor Elena Mazza. She is Professor in iPSC Cell Therapies at the University College Cork in Ireland. And online is present, and you see her there, Dr. Mariana Argentiano, and she is Senior Scientist of Stem Cell Biology at the NCardia Institute in Leiden in the Netherlands. I will introduce the 7 opponents later during the ceremony, because if I mention all those names now, you will certainly forget, so I do that later. And I would like to welcome you here in the aula, and I would like to welcome also all followers of the live stream. And then I would like to present to you the name and of the candidate and his thesis. It is Thomas Hutschalik, and he is defending his thesis in public, and it's entitled In Vitro Modeling of Atrial Inflammation Using Human-Induced Pluripotent Stem Cells. Mr. Hutschalik, may I invite you to give a summary of your thesis, and I wish you success in the coming hour. The floor is yours.
Thank you. Goedemorgen en hartelijk dank. Thank you very much. A big thank you to the highly esteemed opponent, the Prorector, to the members of the committee and the Corona, and a welcome to the guests, colleagues, friends, and my family. I have the honor today to present you my thesis, in vitro modeling of atrial inflammation using human-induced pluripotent stem cells. And I would like to start with an example that you might all be familiar with. You're standing in front of other people, you're giving a speech, and you feel nervous, right? You feel your heart racing, you feel your heart beating in your chest, really pumping in that fast rhythm. And we're all familiar with that, and that's completely normal. But if that beat in your chest would be irregular, out of sync, out of tune, that's an arrhythmia, a heart rhythm disorder, and that's a disease. And the most common one is atrial fibrillation, which is an irregular beating of the upper chambers of the heart. And when you have a fast heartbeat talking in front of people, that's caused by adrenaline. We know that, right? But atrial fibrillation, sometimes we don't know the causes. And one potential reason for atrial fibrillation could be immune cells. And that's what I want to talk about to you today. To give you a quick overview, we'll look into first atrial fibrillation and inflammation, then how you could research that in the lab, then the main part, making little hearts in the lab to study this topic, and then see if inflammation can cause irregularity, and if so, how is that possible? But first, to give you a quick overview, a little bit of background on the heart. You might be familiar with that the heart is a pump. It pumps blood through your body, it's a muscle, but it's also an electrical organ. So the heart consists of 4 chambers. The upper ones, the atria, and the lower ones, the ventricle, who do the hard work of pumping blood through your body, and the atria is kind of the one that supplies the ventricles with the blood to do that. But overall, all this muscle consists of tiny building blocks, the cells. And if you pick one of those cells and you stick a needle into it, you see that it's an electrical being. It has electricity running through its membrane, through its cell body. And you can see that in something called the action potential, where you see different currents running through it. And all of these little cells combined in the heart and combining their electricity form something you might have seen before called an ECG. So all of the electricity in the body. And this looks completely healthy here. It's a normal rhythm. But if this rhythm is disturbed, we get a fuzzy, crazy-looking signal, and that's then atrial fibrillation. So the electricity of all these cells of the whole heart is disturbed. But what we're interested in is to see inflammation in atrial fibrillation. So how does this lead into it? Well, if you look into the heart of a patient, for example, and you look at their atria to see, of course, there aren't only heart muscle cells, the ones that do the pumping and the ones that do the electricity, but there are also immune cells interspersed in between. For example, causing inflammation or fighting bacteria, all different sorts of functions. And of course, there's some other cells there as well. But what we're interested to see is, do these immune cells in between these heart muscle cells cause potentially atrial fibrillation? How would you research that though? How would you find out if these small little building bricks in there influence each other to mess up their electricity? And really important systems that we have been using to find out a lot of things about atrial fibrillations are, for example, patients, right? With patients you can see, do they have inflammation and atrial fibrillation, for example? But with a patient you can't do everything, right? We're all humans, we're not test animals, so you can't do everything with a patient. Another potential way is human biopsies, which basically means taking a piece of your heart, analyzing it in the lab. And using that way, we've seen, for example, there are more immune cells in tissues from patients. But, of course, not everybody of us can give a piece of their heart for research. So then in the end, another big pillar are animal models, which show us a lot of things about atrial fibrillation, and they're really useful. But it's always a whole organism. So you can't study very specific things, because if you test one thing on an animal, you always get the complete organism. So one pillar that I've been using in my research is stem cells, which I want to introduce you to today. You might be familiar with the concept of stem cells. The word stem suggests, for example, if you imagine a tree, a stem cell is the trunk, the stem of the tree, which can become a leaf, a flower, a root. So a stem cell is a sort of beginner cell that can develop into all sorts of different cells. And what we want to do is use these stem cells to turn them into heart cells to then study them in the lab. And how this works is we start again with the adult, the human patient, you, me, and we take some of our cells, some skin cells, a hair cell, some blood, and then we take these cells and reprogram them into stem cells. And this has been really a huge breakthrough by Professor Yamanaka, who got the Nobel Prize in 2012, which incidentally is the year that I started studying, which was one of the reasons I went into biomedical sciences, because the breakthrough was really exciting. The idea here is you take a cell that's already a leaf, a flower, a root, and turn it back into the trunk of the tree. So how this works is you give some factors, some proteins that show the cell, hey, become a beginner cell again, and you get a stem cell. And these are then called, I apologize for the difficult term, human-induced pluripotent stem cells. But this really explains what they are. They're human cells, they're induced, so we cause them to become stem cells, and they're pluripotent, meaning they can theoretically become any cell in our body. And what we want them to become, through differentiation, is a heart cell. And differentiation works that we now have our stem cell, and through kind of the same process of reprogramming, just the other way around, we now tell them to become a leaf, a flower, a root. So we give them proteins, we give them influences that first lead them through this process, kind of like in the human, in the embryo, where from a stem cell, a heart would develop. And we end up with a heart muscle cell that we can now use in the lab. But of course, how do we know it's a heart muscle cell? We need to check, right? And one really exciting part of this is that a heart cell in the lab that we create, it beats. It works like the muscle in the human body. This is under a microscope. You look at it and you see these cells contracting, doing their work, doing their function, being a heart cell, getting electrically excited, and then contracting. And here you see a layer of heart cells under a dish in the microscope beating, and these waves are contractions traveling throughout these cells. But of course, we don't only want heart cells. We want immune cells as well. We want to see the connection between the two. So we also then differentiate it into immune cells. And for immune cells, again, we check, okay, do they have their function? Here, for example, a really common way to see that in the lab is by staining them with colorful fluorescent dyes, where you can see specific proteins on the immune cell. And here you see these guys being stretched out, meaning they're moving around, they're aggressive, they're hunting, they're immune cells, for example, looking for an infection. But of course, we want to combine them. We put them together, and here, for example, you see in green, again with these colorful dyes, the immune cell stained in green on top of these red heart muscle cells. The red here shows the protein, the muscle fibers of the heart cells, and you see them combined, attached on top of each other. And then again, under the microscope, them moving together as one tissue combined with each other. But, of course, 2D flat, that's not really physiological, meaning not very real how it is in the body. Het platteland is not as exciting as, for example, mountainous region as Maastricht, where you combine things together three-dimensionally. And here, for example, we then use the cells that we've developed, put them together around these pillars, these sort of mountains. And then what happens is the cells clump together into a three-dimensional tissue, forming around these pillars and again being able to function like a muscle, contracting, like you would see it in the human body. And if we then check again, you see all these tissues being surrounded by heart muscle cells here in red, the protein, and then interspersed in between, in cyans, the immune cells. But what we're interested then in with this model, with this tissue, is to see, can inflammation cause arrhythmia? These immune cells, do they change the beating of these heart muscle cells? And for that, what we do is, you have, for example, a culture plate where you would place the cells that has electrodes. Here, a grid of these electrodes. Each dot is like one electrode that can measure an electrical signal and see, for example, if there is a beat. So does the cell have an electrical impulse? And then what we do is we place the cells on top and see, okay, is there electrical signal? And then we combine them with our immune cells and see, does anything change? And what you would get out of it is here this electrical signal where each of these signals is, for example, one electrical impulse, one signal, one action of the cell. And as you can already tell here, suddenly something changes. There's a normal rhythm and then something changes, the rhythm is faster. So what we wanted to do is analyze in different conditions what causes these changes. So if we look at just the heart muscle cells on the electrodes, we're looking at irregularity. How regular is the beating? Here, a higher percentage would suggest it's more irregular, it's more diseased. And with normal heart muscle cells, nothing. It's basically 0%. They're beating regularly as they should. But if we combine them with immune cells in there together, close by each other, suddenly there's a lot of irregularity. Something pathophysiological, meaning disease-like, happens. What's interesting is we wanted to see what if we not take the immune cells themselves and put them in there, but just the things they secrete. So for example, the things they sweat out, the things they send out, meaning their fluid, their secretions. And then we didn't see irregularity, suggesting the thing that causes this disease to emerge could be something that needs the direct contact of immune cells and heart cells. But we had our 2D model. We wanted to also see in 3D. In the more relevant model, does the same thing happens. Here we used a camera and a microscope, and then we placed our tissue underneath it, seeing the beating of these tissues, how they contract. And then by recording this and analyzing it through software, we then have these traces showing contractions. Each peak here is the tissue once pulling together, working like a muscle. And you can see, again, this is very regular. Normally paced, every once in a while a contraction. This is a healthy tissue without aggressive immune cells. But then in the inflammation we see, for example, there's a skip beat. It looks irregular, it looks funky. The same thing we saw in 2D, suggesting these immune cells cause some form of disease mechanism. But we didn't only observe irregularity, we also observed that inflammation caused the muscle to become weaker. So for example, contraction, as I was talking about, was reduced. When we see the tissue, how much it pulls together, when it was inflamed, it pulled together less. Important for contraction in the heart is calcium, so an iron. You might be familiar with calcium. But we checked, using again one of these colorful dyes, how much calcium is in the cell during contraction. And here we used one of these microscopes where you could record a lot of individual tissues at once. Each red dot is one little tissue, and when it glows up, we can see if there's calcium in the cell. And we saw when it was inflamed, there was less calcium, less glowing from the dye. And then last but not least, also structural integrity of the tissues was reduced. So for example, we check here for sort of one of the building blocks, the pillars of the cell's collagen, and here we saw that in the inflamed state, this was less. But of course, if we're talking about inflammation, some of you might think about anti-inflammation, anti-inflammatory drugs. You might have heard of cortisol. Some of you might even be taking, for example, anti-inflammatories. And when we treated the cells with these compounds, added them on top, we saw that this loss of contraction was reverted. So by suppressing the immune cells, we were able to restore the normal function and also the rhythm. So irregularity also was reduced, suggesting that really it's inflammation causing these changes and not other things that are not related to inflammation. And of course, suggesting that anti-inflammatories could be an option in some cases against arrhythmias. But what we wanted to see is, how does this happen? What is the mechanism here? And for that, we looked at RNA. Some of you might know DNA, our genes. Basically, RNA is what the genes or DNA send out saying, hey, build me my protein, please be active. So RNA shows which genes are active, which genes are being used. And when we analyzed our inflamed conditions, we saw that important electricity genes were less active, including those that even allow the cell to become electrically activated in the first place. SCN5A here, one of those names for these genes. And when we then added anti-inflammatories, they prevented these changes again, suggesting that this connection might be the mechanism. And here in one of these examples, you see in black the healthy cells having an expression, so RNA activation at, for example, 150. Then in red, when it's inflamed, it's reduced by half, so the gene becomes less active, less RNA. And in green, when we suppress it, it becomes restored, suggesting that this could be a potential mechanism that is the RNA that changes in this condition. And with that, I want to conclude, suggesting that with these stem cell models, we can study inflammation in the heart, in the atria, as I've shown you here, suggest that immune cells can cause an irregular heartbeat, as we've seen, for example, in these different models, and that this platform can be used to study anti-inflammatory therapies, as we've seen, for example, in their effect reducing irregularity. And with that, I thank you very much for your attention and look forward to your questions.
Thank you, Mr. Hutschalik. The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Schoen. She is Professor of Experimental Cardiology, in particular diastolic heart failure at Maastricht University. Professor Schoen.
Thank you, Mr. Prorector. Dear candidate, first of all, I'd like to congratulate you with this excellent thesis and also involve your promotion team in this congratulations, because it's really exciting work that you've done. And, well, as always, with exciting work, there are many questions that keep popping up. So even on my way here, I still had like, oh, I can ask this and I want to know about that. So be prepared. I'll first ask a very general question. I'm really interested in inflammation, I've always been. But one of the things about inflammation is that it's so complex. So can I ask you, in what type of inflammation, what aspect of inflammation are you interested in your thesis?
Yeah, I thank the highly esteemed opponent for her question, and I completely agree, right? Here, the overview, we talk about inflammation and immune cells, but as you've suggested, it's a very complicated network with different aspects to it. In our research, we specifically looked at M1 macrophages, being pro-inflammatory macrophages, and then in the setting that we identified here, it's a very acute inflammation.
Mm-hmm. And would these M1 macrophages in your thesis be the resident macrophages in the tissue, or would they be the invading ones that you mentioned in the introduction?
That's a very good point. It's another degree of distinction in these macrophages. So we have resident macrophages. So for example, in the heart, they're quite common. They're the fourth most common cell type. And then there are circulating macrophages that come through the blood, right?
So which of those are you recapitulating in your tissue?
With some caveat, because they're stem cell derived, the protocol we use is more likely to be resident. So it's supposed to come from a yolk sac lineage derived, while the circulating ones, they're from the bone marrow derived. So they have some differences in that regard. But as there is quite a high plasticity, macrophages, for example, can be M1, but also become M2. So there is distinction in these groups, but also overlap.
Okay, well, that's very clear then. That clarifies a lot. Thank you so much. Because what I really liked about your chapter 3 is that you then compare your tissue that you've created in vitro with real human atria. And you do that by RNA sequencing. And one of your conclusions is that there are lots of differentially expressed genes, and in your myocytes, you induce inflammation type gene expression response.
Yeah.
So what makes you conclude that it's the myocytes expressing those inflammatory genes?
Yeah, so the way we went about it in the protocol is that, of course, in the co-culture, they're combined together, but then the way we did it is we detached the cells and then separated them using bead separation. So basically you have magnetic beads that bind a specific marker on the macrophages. These are then caught in a magnetic filter or a magnetic trap, and you just end up with the cardiomyocytes. The way we also checked it, of course, in the results afterwards in the RNA sequencing, we looked at, for example, macrophage markers, were they present in these co-cultures after separation, and we didn't see that signal, so we concluded that the separation was successful, and the data we have is just for the cardiomyocytes.
That's a real pity, I think, because the immune cells are so interesting. And one of the ways to answer the resident cell types is to really look at those macrophages in the tissue that you separated, and then compare them with the atrial tissue that you also have. Huh? Okay, but well, that's really nice. Thanks for those answers. And I have a very short additional question, if I may. Yes. So you actually pursue the hypothesis that inflammation causes atrial fibrillation, right?
Yeah.
But in your introduction, you also raised the possibility of the opposite,
yeah.
that atrial fibrillation causes inflammation. How would you address that hypothesis with your current models?
Yeah, I think there's obviously a connection of atrial fibrillation and inflammation that is not causative. We know that, for example, risk factors for atrial fibrillation are things like older age, obesity, and obesity, for example, is connected with inflammation levels. So obviously there are also parallel factors going along each other where not necessarily inflammation causes atrial fibrillation. The reason is we chose this specific setup was really to show that it's this initial causes that has atrial fibrillation, and how we were able to see that is temporarily, we have activation of the macrophages in the co-culture, and then a day after, self-emergent irregularity happened, and then of course looking at just activation factors, just supernatant, and we were able to see that from this time frame that only the combined culture, macrophages, activation factors, heart cells, caused this emergent irregularity type, which at least to me seems like it is causally connected.
Sure. So that's, again, like pursuing the hypothesis that inflammation causes AF. Huh? That's exactly what you did. So could you turn it around? Could you also use your model to show that AF, or the irregular heartbeats in your myocytes, can activate your tissue-resident macrophages?
I think there definitely would be opportunities. I mean, one common way, for example, to induce atrial fibrillation is through burst pacing, so giving cardiomyocytes rapid beats, which then, for example, could induce an atrial fibrillation type, and then in that co-culture see, does it change the subtype or the profile of the macrophages? I think that could be potentially possible. Since we didn't pursue burst pacing to induce atrial fibrillation, I can't definitely say that it works with the model, but it is definitely interesting.
Well, yes, that would be so interesting. And also an opportunity would be to combine other factors that are playing a role in cardiovascular disease, like the renin-angiotensin system, for example, because macrophages do possess angiotensin receptors. So that would also be really nice, because angiotensin, I don't know if you're aware, maybe it's a little bit of a lecture, but actually causes macrophages to become purely M1, while the exposures that you have are also causing M2 differentiation.
Yeah.
We
have to move on.
We move on. Thank you so much for these answers and listening as well. I give the word back to the prorector.
Thank you.
Thank you, Professor Schoen. The opposition will be continued by Professor Pijnappels. He is not present here in the aula, but you see on the screen. He is Professor of Cellular Electrophysiology at the Leiden University Medical Center. Professor Pijnappels.
Thank you very much, Rector. So, dear candidate, first of all, congratulations on producing such a nice thesis on an important topic, the role of inflammation in atrial fibrillation. So, my first question concerns your main aim to study whether there is any role. And so, also based on your nice presentation, I guess the answer is yes. But could you please explain to me, as you probably know, there are more factors involved that we know of in causing atrial fibrillation, like fibrotic and adipogenic remodeling. So if we now also include inflammation into this equation, how would you see the weight of inflammation as compared to the other pro-arrhythmic factors in causing atrial fibrillation?
I thank the highly esteemed opponent for their question, and I completely agree. We know with atrial fibrillation that it's a combination of factors always. So usually it's a trigger and a substrate that needs to be present for atrial fibrillation to emerge. So ectopy, for example, itself does not necessarily lead to atrial fibrillation. So then the question is, of course, how does inflammation lead into this? And I think one interesting example here, for example, is the setting of post-operative atrial fibrillation, for example, where you have surgery, you have sterile inflammation, and then patients, for example, get this setting of atrial fibrillation afterwards. So I would suggest from our research in the smaller models that the changes in electrophysiology in cardiomyocyte lead, for example, to an increase in automaticity, at least in our model, which could, for example, be a cause of arrhythmia to that degree. Some other models, for example, that looked into sterile inflammation, for example, in animal models, where they induced atrial fibrillation through burst pacing, saw that after burst pacing in dogs that had this sterile inflammation, there was a higher likelihood of induction of atrial fibrillation, which also could suggest that atrial fibrillation or inflammation also affects the substrate as well. So a little bit of both sides potentially making automaticity.
Sorry to interrupt you, but so I was more interested in the particular hierarchy. So is the one more pro-arrhythmic than the other? Do they need one another? Does it depend on the scenario? So the absolute weight of inflammation, fibrosis, adipogenic remodeling. Can you please speculate a little bit about the hierarchy?
So I could, for example, suggest the computer modeling we did, where, for example, we looked at these changes we identified spread out in the heart or centralized in the heart. So these genetic electrophysiological changes, which then emerged atrial fibrillation after tachycardia. And in these models, there, for example, was no fibrosis, no other remodeling besides the changes we observed, which could suggest that these inflammation changes by themselves might lead already to atrial fibrillation, not necessarily having additional factors like rapid ectopy or like fibrosis, which potentially could mean that this is an initiating cause by itself.
Yeah, thank you very much. So my next question concerns chapter 5. There you nicely describe a 3D model based on the combination of culturing myocytes, fibroblasts, and macrophages. And I have now two more technical questions. So you could not electrically pace due to the fact that those cultures were spontaneously active, and the tissues were also too small to be optically mapped to actually visualize the wave propagation. So my question is, how do you actually exclude the possibility that the beating irregularities as observed in your model are not so much related to pro-fibrillatory mechanisms, but that there is actually something happening to the mechanisms involved in spontaneous activity?
Regarding that aspect, I completely agree that there are some limitations to the tissues, for example. They are quite small, so we did try to do some optical mapping, but of course, because of the size, this is not very informative that much. So things like rotors, etc., are not very likely. Also, we didn't publish that data, so it was more a test run with the optical mapping. Things like a lot of ectopic beats happening, we didn't see that much either. So it seems to me that the mechanism here is related to automaticity, as we've seen, for example, in the sharp electrode recordings, where we had things like the action potentials having alternance, the maximum diastolic potential being less negative, aspects like that, to me suggesting that the individual cells have perturbances in their action potential, which then leads to the emergent arrhythmia.
And yeah, so thank you for your answer. But if I would phrase it differently, so if you would be able to inhibit the spontaneous activity, and you could electrically pace the cultures at different frequencies, to what extent do you think that the outcome would be different? So to what extent does it actually depend on spontaneous activity?
Yeah, I think that's definitely an interesting point, right? Because the stem cell model, it depends on this automaticity. We've been using that, so we haven't done pacing to not have this effect be present. And we try to investigate if there is some degree of, for example, funny current that is in the stem cells. And for example, we didn't see transcriptional changes in HCN4 in the different conditions, suggesting that the funny current channel isn't upregulated or downregulated. So it might not be that this specific pacemaker current is really necessary for the effect we see. But besides that, it could be that if the cells were paced, the effects we see would still emerge, as for example, we suggest the reduction of connexins, for example, less interconnectivity between the cells. I think that effect would still be present if we would be pacing. The inhibition of the sodium-potassium pump, so restoring the maximum diastolic potential, that effect I think would still be present in pacing. But it would definitely be an interesting aspect to look at if we inhibit this automatic rhythm of the tissues and look at a pace rate, if there still is that effect. Yes.
Yeah, thank you. Well, probably my last question for the sake of time. So it's about your about the tissue composition. So you put together those cardiomyocytes, fibroblasts, and the macrophages, and you do so in a particular ratio. And could you please specify in a little bit more detail, how did you come up with this particular ratio? Was it trial and error, or is there actually reasoning involved? What about this ratio?
Give an extremely short answer.
Extremely shortly, we looked at the physiological relation of cardiomyocytes to macrophages, which is sort of in that ratio that we had, so 20,000 to 5,000, 1 to 4, which is also what you would find in the body. And then the fibroblast ratio is a lot also based on practicality for the tissues to form.
Oh, well, thank you for your answers, and I give the word back to my director. Thank you.
Very much.
Thank you, Professor Pijnappels. The next opponent is Professor Ravens. She is Professor of Cardiovascular Pharmacology at the University of Freiburg in Germany. Professor Ravens.
Thank you, Mr. Prorector. Mr. Candidate, I would also like to congratulate you on this applaudable amount of data you have collected in such a short period, which a PhD comprises. I would like to continue on the discussion you had with Professor Pijnappels about the irregularity of the rhythm. And I have a rather technical question. In your chapter 3, you talk about the percent change in irregularity. And irregularity is defined as something, beats per minute, which are then taken the standard deviation. For this, you need a certain period of time to record. I did not find any information on how long you will record and count the beats within this. And please explain to me how you define irregularity.
Yes, of course. I thank the highly esteemed opponent for her question. Firstly, so the recording time, we usually do 10 minutes. So in that time, we end up with, let's say, 600 beats. So these are then averaged to receive the average beat rate, which then, as you've of course stated, we then use the standard deviation to find a ratio of standard deviation to beat rate. Yes?
May I interrupt exactly here? I did not understand how, when you count beats within a certain time window, you can derive to a standard deviation, because you have an average value. Where is the standard deviation? How do you calculate the standard deviation for this?
So, for example, there in that frame, I'm trying to look if it is stated in the methods. So,
in your figure in chapter 3, you give a short trace. You also showed it nicely in your presentation, where you show a change in frequency, a sudden change in spontaneous frequency of your beats, and then you come to the definition of irregularity, which
exactly.
I did not understand.
No, I completely agree. I'm happy to explain. So, of course, the beat rate is the mean, but then for the standard deviation, each time frame of a beat, right? It's the beat period, so the time between these beats is converted to an individual beat rate.
Oh, so you take the interval between the beat,
Exactly.
and you count the different, or you take the standard deviation from the different intervals between beats during those 10 minutes.
Exactly.
Now I get it. Okay, thank you. Thank you very much. This really bothered me a little bit.
I apologize.
This part. Can we switch gears a little bit? In your method section of chapter 2, you describe that you differentiate your cardiomyocytes in the presence of a very commonly used drug, fasudil, or fasudil, however you pronounce it in English.
Yeah.
That is usually used as a muscle relaxant.
Yeah.
So what did you use it for in your differentiation protocol? Why do you have to have it?
Yeah, so this is based on experiences prior. So a lot of course, my differentiation protocols, they build on top of technology developed by Nkardia. And here, fasudil specifically acts as a ROCK inhibitor. So basically, if you would not add this ROCK inhibitor during these early stages, when you seed the stem cells, they would go through apoptosis. So the ROCK inhibitor here, fasudil, leaves the cells alive for us to culture them in the dish.
Could you do this with any other apoptosis inhibiting drug?
So we do use other ROCK inhibitors, for example. This is based on experience at Nkardia that I had been going through. So other ROCK inhibitors might be an option. Fasudil is just the one that has been established there for that protocol.
Mr. Prorector, do I have time for another question? A very... Well, I don't really have a short question, so we leave it later to the discussion. Thank you very much, Mr. Candidate.
Thank you.
Thank you very much, Professor Ravens. The next opponent is Professor Leon Schurgers. He is Professor of Biochemistry of Vascular Calcification at Maastricht University. Professor Schurgers.
Thank you, Mr. Prorector, dear candidate. Also, I read your thesis with great pleasure, and I read it completely because it's a topic that is a little bit aside of my topic, but it's very close to my heart. And I was really pleased that you use human-induced pluripotent stem cells and stepped away and deviated from the mouse models that normal people use, because that is only bringing good news to all the mice in the audience and not to the humans in the audience. In these congratulations, of course, I want to also extend this to your supervisory team. When I received your thesis, I was a little bit surprised by the size of the booklet, which is a little bit different from the Dutch standard. And as you like German quotes in your thesis, I did not understand why you not had chosen for the one from Goethe, in der Beschränkung zeigt sich der Meister. But anyway, we're here to have an academic discussion. So my first question is, you did your PhD between industry and academia. What is your next path? Is it industry, or do you choose academia?
It is, of course, unfortunately, in this setting, it is industry. So, yeah, after the completion of the PhD, I will be pursuing an industry position. But the research actually itself stays close to me. We still have publications that are currently going through review processes. So there is more of this to find its way into scientific journals. And of course, stem cells and cardiology is an interest of mine. So I definitely am not adverse to stay close.
I think it's also a new era where industry and academia closely work together.
Yeah.
But going into your thesis, I want to ask one of your paranymphs to read proposition number 5, please.
Sure. Proposition number 5. Inhibition of this inflammation could be a target of prevention or treatment as identified in vitro.
So you have to be aware that if you put on a proposition, you have to defend it. So what is disinflammation and what target?
Yeah, of course, regarding, I think the highly esteemed opponent for the question, regarding the inflammation, the macrophages, their activation, their passage from M0 to M1, so this pro-inflammatory action on the cardiomyocytes, and then the target here is more diffuse in a way. I think it's framed in a way that leaves it open to potentials, as we've been using, for example, hydrocortisone, dexamethasone, glucosteroids, which are very broadband. They affect a magnitude of things in inflammation. So we kind of use them as a general suppressor of inflammation overall. Yeah. And there is some data in meta-analysis that suggests, for example, patients that underwent cardiac surgery and receive steroids, that they have a reduced incidence of post-operative atrial fibrillation.
But you're going to, so this inflammation is meaning the M1 macrophage phenotype.
Yes, exactly.
So fibrosis is also one of the inducers of atrial fibrillation. Now, this is normally caused by an M2 phenotype because that's the reparative macrophage. Why did you not choose to compare M1 and M2?
That's a very good question. Of course, I think here in our setting, we really wanted to narrow down the question on the acute setting. Does this initial burst of inflammation, could this cause a change in electrophysiology? Then going beyond that into more the second stage, the later stages of inflammation, where there is a more M2 phenotype, a more reparative mechanism. Unfortunately, that was not in the scope of the work itself. We tried to lead towards fibrosis as a potential investigation. So we looked at collagen deposition with the fibroblasts, where we, for example, saw some changes initially. But this is definitely an area of interest as
And you stay in industry, so you have this industrial, let's say, glasses on. So if you think of drugs, then you can think of, okay, I want to limit the inflammatory response, but you can also think of, okay, changing the M1 to the M2 by resolvins.
Yeah.
Do you have any idea of what kind of drugs you could use to switch the macrophage phenotype from M1 to M2?
Regarding that, since we didn't move so far into M2, there specifically this change, I wouldn't necessarily be aware of compounds that are available. Of course, in the differentiation, we observed different cytokines that lead towards M2, for example, IL-10, which is used in that process. But regarding the specific switch from M1 to M2, unfortunately, there I'm not aware of potentials.
And so if you look at the tissue, you studied the M1 phenotype, and you really did great work, so don't misunderstand me. But the question here is, where does the M1 tissue macrophage phenotype come from? Is that not the stage where intervention should be targeted on?
Yeah, I think specifically looking at this M1 inflammation, I think there's one of the major limitations as well in the thesis. So for example, how we induce the type is, over a few days with GM-CSF and then activating it with interferon gamma and LPS, which suggests, for example, a bacterial infection, right? So the setting here could be acute inflammation related to bacteria, which is a very specific setting. So it is acute, it is pro-inflammatory, but then, for example, how this could happen in the physiological setting, it's less clear from this specific model. Yeah.
Mr. Prorector.
Very small one.
Could the impairment in conduction of cardiomyocytes also be caused by the number of macrophages in between the cardiomyocytes, thereby limiting current transfer?
Yeah, that's definitely an interesting question that we thought about as well, right? We know that, for example, fibrosis inhibits this conduction. Could macrophages have the same effect? So there we really, in the protocol, looked into different controls. So we included controls where we have cardiomyocytes with, for example, macrophages that are not activated. So then using, for example, immunofluorescence staining, we looked at these ratios of cells were similar. So just by the introduction of cells between the cardiomyocytes, we didn't see arrhythmia, suggesting that it's more the activation and not just the presence of the macrophages.
I thank you for all your answers and keep continuing this work. I give the word back to Mr. Prorector.
Thank you, Professor Surgers. The opposition will be continued by Dr. Nabben. She is Assistant Professor of Heart and Metabolism at Maastricht University. Dr. Nabben.
Yes, thank you very much. So first of all, I would also like to congratulate you and your supervising team, also online, on this beautiful piece of work. I think it's a very nice combination of there's in vitro work, maybe even a bit of ex vivo towards ex vivo, in silico work. So a lot of work that you've done. But of course, I also have some questions. And since your paranymph did such a great job, I would also like him, or maybe the other paranymph, to read proposition number 7 in this case.
For stem cell-derived models to offer relevant data, sufficient number of donor lines must be available to reduce the impact of individual outliers. This includes the representation of sexes, global populations, and tissues of origin in the utilized lines. To achieve this, scientific collaboration and robust procedures around informed consent are paramount.
Thank you. So, in relation to that, there you basically acknowledge, well, collaboration is important, but you also talk about numbers of donor lines and yeah, I guess also numbers in general. So, since that is so important, when I then go to your to page 24 in chapter 2, there's a lot of figures in there. And if I then look at these figures, I see, for example, well, I see a lot of, well, sometimes I don't see dots, sometimes I see dots. For example, in figure 6, I counted sometimes 20 dots, sometimes 32, I think. And then in figure E, I count 2 dots and sometimes 3 dots.
Yeah.
What are these dots? What are these numbers?
Yeah, I think that's a very important distinction. So obviously, talking about lines, in this chapter, we're looking at 2 donor lines. Later on, we're looking at 3 donor lines. So basically, these are the different organisms that are represented. Then here, depending on these individual dots, for example, looking at subfigure C, these are then individual cells. So this is then in sharp electrode recordings where we get the signal from one individual cell. Here then, for example, in E, it is the individual well, so the cells that are placed into the dish in one well. So one data point is the result from one well. So here usually it's always separated by the technical replicate, so individual cells, individual dishes, individual tissues.
Yeah, so indeed, the technical replicates, sometimes it's the biological replicates then also, so the donor line. So it could also be basically the different differentiation lines, numbers, right? So in an ideal world, what do you think is the best? Or what would you aim to or suggest people to aim at?
Yeah, I think here for the initial work, I think 3 is often the magic number, right? Using 3 different donors, using 3 different differentiations to reduce the effect of individual processes, right? Different differentiations can lead to different cells, which show different electrophysiological status. Different donor lines can have different results. And we see that in the data as well. I think even with the lines, I was able to see that they often respond differently to the differentiation as well. So for example, one line was very well at producing macrophages, the other one was very good at producing cardiomyocytes. So there, even in this small array of potential donors, you see variation. So I think there it's definitely important to at least have 3 different lines. But then, of course, one major consideration when it comes here, to example, to immunology. In the RNA sequencing, MHC comes up, major histocompatibility complex. And there we know that different individuals can have different backgrounds when it comes to immunology. So even if you would just want to represent all the phenotypes of different MHCs, you would need several lines. So in that regard, I think this is more seen as a proof of concept for the potential and not necessarily an accurate representation of humanity.
Yeah, I think I agree with that. But also in relation to that maybe a little bit, because also in the proposition 7, it was about representation of sexes.
Yeah.
If you would want to study sex differences, how would you do that? Because now I think you had a mix, right? You had some female and...
Yeah, I think that was intentional from our side to have both sexes represented, but of course in 3 lines, there's not a large degree between sex differences. So if we would see differences here between the line, we would have 2 lines on one side, 1 line on the other side. So you would impossibly be able to state this is a sex difference. But of course, we know that sex can influence, for example, atrial fibrillation as a risk factor. So here then again, it would need to be increase the number of comparisons, increase the number of donor lines to accurately say, okay, here we see an emerging difference between male and female lines.
Would the magic number 3 would count again? Like 3 male, 3 female donor lines?
I think in that case, I would be a bit more skeptical there. I think the rule of thumb is the more you're able to compare, the better. That's especially why I wanted to make that point about collaboration, right? Several labs have different lines available that have informed consents, and they're combining the power, combining the ability to have access to these lines really can scale the impact of the individual work.
Okay. Is there still time for a question or?
Yeah, you have time for one question.
Okay.
Yes.
Yeah. as you might have heard, I study cardiac metabolism. So I'm also interested in, well, metabolic maturation in a way. And I did not really read that in your thesis.
Yeah.
Do you know, are they metabolically just as mature when you compare the cell lines? Did you look at metabolic maturation, or do you have any idea about it?
So regarding metabolics, we didn't really look into that. So especially in chapter 2, we discussed the different paths to differentiation. And for example, metabolic selection is one mainstay where you push the cardiomyocytes more towards a matured metabolism by depleting sugar and having them have that fatty acid oxidation. For the differentiation itself, one of the goals was to not have metabolic selection, right? Mm-hmm. So we observed that more as a hurdle. But then looking into how matured they are, how their metabolism is unfortunately not there, I couldn't make any statements about the lines.
But they are the same maturation state, you think? In general, I mean, not metabolically, but in general, because if you compare lines, they should be the same age, basically.
Yeah,
right?
I think for us, of course, timelines, they're all the same. So there we always use the same processes, the same protocols. If you look at the electrophysiological parameters, they mostly align. So they have similar maximum diastolic potentials. They have similar upstroke velocities. So there, at least, the maturation seems to be comparable.
Okay, thank you.
Thank you.
Give the word back.
Thank you, Dr. Nabben. The next opponent is Dr. Matthijs Blankenstein. He is Associate Professor of Pharmacology at Maastricht University. Dr. Blankenstein.
Thank you very much, dear candidate. I also enjoyed very much reading your thesis, and I would like to congratulate you with the completion of the very nice work, and of course include your team that contributed also. And yeah, as a pharmacologist, I would like to take you to chapter 3, where you show that you can use glucocorticoids, particularly dexamethasone and hydrocortisone, to reverse the effects of the M1 macrophages that you add to your cultures. However, you also used ibuprofen as an anti-inflammatory agent.
Yes.
And ibuprofen did not reverse the phenotype that you saw. So, taking that in mind, do you think that the title of the paragraph, anti-inflammatory compounds alleviate inflammation-induced electrophysiological phenotypes in ACM, on page 44, is a very well phrased, or would you like to reconsider that?
Well, I thank the esteemed opponent for his question, and I do agree. In the direct instant, the generalization might be too generalized. So, of course, yes, we observed that with this immunosuppressant, the steroids, there was a reduction in irregularity, but of course, with ibuprofen, that was not the case. Mm-hmm. So you are correct.
Yeah. Okay. Well, having said that, you mentioned in your discussion and also in the general discussion that you found clinical trials where actually treatment of patients with glucocorticoids has a beneficial effect on AF. Well, I also looked a little bit in literature, but I also found a lot of studies where either there was no effect or there was actually a detrimental effect. Particularly high-dose dexamethasone can have as a side effect the induction of AF. So when it comes to clinical application, do you think administration of glucocorticoids would be the way forward to treat AF?
I think it's definitely an important point that glucocorticoids, for example, especially dexamethasone as a very powerful immunosuppressant, is likely not to be, especially in this case of AF prevention, really a choice. So I can't see a clinician thinking, I want to prevent atrial fibrillation, so I'm going to give someone doses of dexamethasone. I think that's not reasonable to assume. In this specific case of post-operative atrial fibrillation, as there is some data there that suggests there could be a connection, there could be a potential benefit, but overall, because glucocorticoids have a lot of side effects, right? Osteoporosis, Cushing's syndrome. They are not really useful as a targeted therapy to prevent atrial fibrillation. So I think here it's more the idea to show suppressing the inflammation in general prevents the effect in vitro. And then to move beyond this in a clinical setting, there would have to be some form of targeted treatment that doesn't have this side effect portfolio of glucocorticoids.
Well, I'm happy you used the word targeted therapy because that's exactly the direction I would like to take. Do you have any suggestions of therapies that could be more focused and could be tried in this context?
Unfortunately, in electrophysiology and anti-arrhythmic treatments, often the use case is suppressing a channel, right? Blocking it. But the electrophysiological changes we observed are all down-regulation mostly. So, of course, it's difficult to restore the effect of the sodium-potassium pump or restore the effect of connexin. So,
but if I may interrupt you, if you think along the lines of inflammation,
yeah.
Would there be any options there?
Exactly. So this is then really, I think, the area, the alley to find a potential to counteract this. Unfortunately, I think in the work going on this side where the macrophages affect the cardiomyocyte that causes then these changes transcriptionally, there, unfortunately, this was a bit out of the scope of the work. So currently, I don't have very strong hypotheses to say, okay, this specific axis of interaction here could be druggable to then prevent the transcriptional changes.
Well, actually, a potential answer maybe on the next page, 45, where you show IL-6 release. There is actually a clinically registered drug that blocks IL-6. Would that be an option to try?
Of course, there's, for example, some antibody drugs that target IL-6. I think for us here, IL-6 was the choice as a marker of inflammation and not suggestible as part of the mechanism itself. So I think here we wanted to use an ubiquitous inflammation marker that shows that hydrocortisone suppresses the function of the macrophages. So I wouldn't feel confident to suggest that suppressing IL-6 would have actually an effect on the arrhythmia we observed.
Okay, well, thank you very much for your answer, and I give the word back to the prorector.
Thank you.
Thank you, Dr. Blankenstein. The seventh and last opponent is Dr. Frans van Nieuwenhoven. He is Associate Professor of Physiology.
Thank
Dr.
you, Mr. Prorector. Dear candidate, I also would like to congratulate you and the supervisory team. It's a beautiful thesis, very interesting. Also, your presentation today was very nice and very clear. I read it with the thesis with a lot of interest, specifically the part on the engineered heart tissue and how you develop these models. You actually refined the differentiation protocols for all the different stem cell types, for the different cardiac cell types, and as you might have expected, I'm interested in the interaction between the cells. So I focus my discussion with you on the importance of the paracrine signaling between the cells, and also with a focus on cardiac fibroblasts and the extracellular matrix. Now, first in chapter 3, you also showed it in the presentation. You in the co-cultures of the cardiomyocytes with the M1 macrophages, you see that there are electrophysiological abnormalities. And then somewhere in the discussion, and you compared the direct interaction between the cells and also the conditioned media. And then you say, from these results, you hypothesize there must be a direct interaction between the cells. I would like to ask you to speculate on the mechanism of this interaction. So how do you think this would work?
Yeah, I thank the esteemed opponent for his question, and I think that's really the crux, right, to identify what is this connection, because we do know there are, for example, cytokines that can increase AF inducibility. There are other groups that, for example, studied mice where they see IL-1 beta increases an arrhythmic effect, but of course we didn't observe that. The supernatant didn't have a pro-arrhythmic effect. When we look at the bigger picture, when we went to RNA sequencing, we, for example, saw in the 3 groups of the healthy cardiomyocytes, the ones treated with supernatant and the ones in co-culture, that the effect of the co-culture was much larger than the effect of the supernatant treatment. So there seems to be a plethora of things additionally than just the cytokine secretion. So there needs to be some effect of this interaction. And when we look at the specific gene ontologies that were upregulated, these were things like antigen binding, major histocompatibility complexes. For example, we know these are useful for the cells to communicate with each other. So cardiomyocytes communicating with immune cells, but also immune cells communicating with each other. So it could be that one of these cell-bound proteins, one of these cell-bound receptors, once they interact with a macrophage, then starts, for example, an inflammatory cascade, which then leads to potentially changes in the transcription regimen, which then leads to these electrophysiological changes. Which specific...
I agree with that, but I also wanted to ask, because I have other questions and limited time.
Please.
Do you think there are alternative explanations for the fact that you do not find this strong effect in the use of the conditioned media? So you say these cytokines are in there and they don't.
So there are some limits to the use of supernatant, for example. As you've suggested, there are paracrine factors as well, right? Things that are very in close connection that might not necessarily be cell adhesion or cell attachment. For example, nitric oxide, which is something macrophages secrete, but which has a very short half-life. So already transporting, for example, the supernatant from one well to another might reduce that. So this could be a potential target, something that I haven't investigated myself. But it seems at least that looking at the transcription changes, it should be something that, for example, luminal side of the membrane was also one of the gene ontologies highly upregulated that we also found in clinical samples. So it could be something where really cell-to-cell contact is a major influence.
Do you think that maybe local concentrations of cytokines, that cytokines produced in the vicinity of cells, maybe in combination with matrix, might be different than using a conditioned medium where they might be diluted? And
I could see that these interactions might play a role in the 2D setting where we only have macrophages and cardiomyocytes. There, of course, extracellular matrix is not a major factor, I think, in the in vitro model. In the tissue, this might be the case, that this triangular interaction might increase some effects there. But regarding cytokine local overexpressions or local increased concentrations, there I would have to speculate if that has a specific effect.
Mm-hmm. Okay. I would like to turn to the matrix, which you used for the 3D engineered heart tissues. And can I finish the question? No.
Mr. Hutschalik, the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis, and in particular the quality of your defense, and I request you to remain here and await our return.
Thank you.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in, it's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Mr. Hutschalik, the committee here present, not only here but also online, has assessed the quality of your thesis and the quality of your defense. And in view of its positive verdict, and of course taking into account your previous qualifications, the committee has decided to grant you the degree of doctor. And Professor Schotte is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law. I give the floor to Professor Schotte.
All right.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Thomas Günter Alexander Hutschalik, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
We'd like to invite your father to take a picture.
Please be seated.
Dr. Hutschalik, dear Thomas, this was an excellent defense, really. You did a fantastic job, despite the fact that there was some resistance to make your presentation easier, and you in the end really gave a fantastic presentation. And I'm very happy for you, and I really enjoyed this process overall with you, and the official laudation is now going to be given by Elena.
Thank you. Ladies and gentlemen, thank you all for being here today. It is my personal honor to be giving a talk for Thomas, a scientist, strategist, public speaker, and community organizer whose light shines brighter than this aula we're starting in today. My name is Elena. I was Thomas's PhD supervisor from 2020 to today. And I'm so proud to be here today and also happy to be finally free. I'm currently a professor in Ireland, so I flew in for this ceremony, which of course I was very happy to do, to celebrate Thomas. While supervising Thomas, I was of course employed in the Netherlands at Incardia, where Thomas was based for the first 3 years of his PhD. And at this point, I would like to thank Incardia for sponsoring my flight to be here, for my bonuses, which I no longer have as an academic, but of course, joking aside, for providing a very professional, scientific, excellent environment in which Thomas did his PhD. At this point, I would also like to thank Uli, Mariana, and Oscar Bartulos, who is not here today, for their support that they gave to Thomas, and Stefan Bram, the co-founder and then CEO of Incardia, for contributing ideas to his project. I hired Thomas as one of the first things I did as director at Incardia. I interviewed many candidates, and Thomas stood out for his can-do attitude and radiant positivity. he wasn't maybe the most qualified or didn't have the most hands-on background in scientific techniques, but I was convinced that he had the personal qualities, the perseverance, to be able to endure the hardship, the long hours, dedication, the rejection, and of course the bad late-night meals that are needed to complete a PhD. So, ladies and gentlemen, today we know that I was right to hire him. Thomas arrived in the lab full of confidence, ready with enthusiasm to tackle his PhD. Of course, it wasn't long after that I had to file a safety report because Thomas stabbed himself with a glass vial. Parents, you're here. No reason to be alarmed. I think the most damage was to his ego. Through this and other incidents, Thomas eventually put his feet down and worked really hard. He was known for working into the late hours of the night, wearing his headphones into his own zone, just working away. He was so focused, but also very ambitious. In fact, he had a bet with another scientist at Encardia that he would publish his first paper within a year. He lost. Of course, it took him 4 years to publish a paper, which is the normal time, by the way. It takes someone to publish a paper in our field. Nevertheless, Thomas maintained his positivity. In fact, his can-do attitude was so infectious, it spread in the office to everyone. His big speeches, full of big words, were inspiring, and his colleagues do miss that, as they tell me. What they don't miss is the mess he left behind in his path, the piles of dishes and papers and empty cups of noodle soup that he too often had from the company kitchen. So Fed by Encardia was definitely a logo that he fully embodied. Of course, it wasn't all cups of noodle soup. Thomas built a great community of colleagues around him. They often cooked meals for each other and had very great collegial collaborations. This community also helped him and other PhD students through very difficult COVID times that Thomas did his PhD during. In fact, one of the colleagues lived across the street from the company, and for a few weeks, Thomas and other PhD students would stay there so that they could sneak into the lab without a fear of being caught by the COVID police, so that they could go in and do their experiments. So, all puns intended, but while living out of a suitcase, he managed to dodge the authorities, ensuring that his vital cardiac research never missed a beat. Thomas's PhD work bridged the gap between scientific discovery and real-world impact. Through a prestigious EU-funded PhD studentship, he developed cutting-edge disease models for atrial fibrillation, a devastating cardiac arrhythmia. His work enhanced our understanding of how the disease develops and suggested new ways in which it can be potentially treated, helping patients. He contributed to the development of organoid systems, mimicking the heart's function in a petri dish, which even to this day, despite decades of working in this line of work, I find fascinating. He contributed to the development of advanced pharmaceutics. His work has been published in a peer-reviewed journal, demonstrating its validity and merit. Thomas's excellence has also been recognized through numerous awards, including winning the Falling Walls competition for his beating heart video, which was an achievement showcasing his scientific excellence, but also his gifts for communication and creativity that made complex science accessible and engaging. He additionally received poster awards for his research at international conferences and was an active member of the Early Career Advisory Committee at ISSCR, the biggest worldwide community for stem cell research. Through this, he organized the first ever ISSCR local symposium right here in Maastricht, and for this work, he received the ISSCR Public Service Award in 2025. It is fair to say that Thomas exemplifies the qualities of a gifted communicator who inspires collaboration in others and thrives with theatrical, radiant positivity. His journey inspires the breakdown of barriers between disciplines, between research and application, and between cultures through his travels. He has left an incredible mark on all of us who have worked with him during his PhD, and I'm positive that he will leave an incredible mark on the world of human health through the rest of his journeys. Ladies and gentlemen, please join me one more time in congratulating Dr. Thomas Hutschalik.
Esteemed Dr. Hutschalik, dear Thomas, it's my great pleasure to congratulate you with your doctorate, and I do that also on behalf of Maastricht University. And I would like to look back with you to your presentation, your thesis, and your defense. First of all, I would like to compliment you with an excellent summary of your thesis, giving and explaining the background, the research questions, the performance of studies and the outcomes. And this presentation, this summary, is meant for non-experts. And I really think that many people who are non-experts have very well seen and heard what you have done. It was very, very clear. And Maastricht University also appreciates the fact that the scientific work is presented to the general public. And it was very good. With respect to your thesis, it was a well-written thesis, interesting and relevant topic, and an excellent array of experiments. And it demonstrated hard work. With respect to your defense, we are impressed by your clear answers, by the relaxed way of presenting it and defending it. And it was convincing, it was honest, open, and it also represented the state of art. And I think that to complete this sort of short laudatio, that you are very knowledgeable in the field that you have studied, but also that you have demonstrated that you are very well able to independently perform scientific work. And I do not only congratulate you with this result, but also your three supervisors, Professor Uli Schotte, Professor Elena Mazza, and also Dr. Mariana Argentiano. Congratulations with the results that we have seen and heard today. And I would like to include in my congratulations, of course, your family, your parents sitting here on the first row, and I understood also your brother, who had an important role in this academic ceremony. So congratulations. And I would also like to include in my congratulations the people present here in the aula, and I think that also in the live stream that many of your colleagues and other family members and other people that are interested in your thesis have enjoyed viewing this and hearing this. I would like to thank all members of the degree committee and of the thesis assessment committee, and I would like to thank you for critically reading the manuscript, but also for your interesting questions. And I think that most people have enjoyed the discussion that we have. So thank you very much, and I would like to especially thank the external members of the committee, Professor Pijnappels, present here on the screen, and Professor Ravens, who are external members of the committee. Maastricht University appreciates your input. Thank you very much. I would like to give the opportunity to your supervisor, Dr. Mariana Argentiano, first the word to congratulate you.
Dear Thomas, it was excellent to see your thesis today. I can just say how proud I am of you, how much you have accomplished. I've seen you grow through these years. And of course, I have dodged the bullets for you and stopped the bullets for the mess that you left, as Elena mentioned, as well as the amounts of data that you store in the computers and people couldn't store their own data. But they still miss you, and we are all happy to see your progress and how you have grown into a great researcher and person as well. I think that this defense was a great reflection of your hard work, your resilience, your curiosity as well. So you have done a great job, and it's been a privilege to work with you and to be part of your journey. So the only thing that I can say is congratulations. It's a well-deserved success, and I cannot wait to see what comes next, what future, bright future comes to you.
Thank you, Dr. Argentiano. May I invite Professor Pijnappels?
Yeah, dear Dr. Hutschalik, well, congratulations on obtaining your PhD degree. A great presentation you gave, and I also very much enjoyed our discussion, so thank you for that. And I also would like to wish you all the best with the next steps in both your life and career. Once more, congratulations.
Thank you very much. Then I have, before I close this academic ceremony, I would like to ask the people here in the aula to go after the closing of the ceremony to the rafter. You find the rafter quite easily if you leave the aula and you go to the elevator and you make a turn right, you come to the rafter, and there we will see you for the reception for which you are invited. I would like to invite the committee to, after closing of this ceremony, to take a picture with our external members online on the photograph, and then we take after that another photograph in the stairs in the hall. And I would like to invite your parents and your brother to stay with us, because we want you also on the picture. Hereby I close this academic ceremony.
All right, please.
Thomas, if you would like to step forward and to stand here with your Paralympics, please take the diploma with you.